Now we're going to talk about a whole nother group of worms, the actual worms. So what we're going to kind of do is, the first thing we're going to do is we're going to kind of cover cryptic species, like what are cryptic species, why are they important, and then we're going to introduce the actual genera, so Stylaria. For the second, big major portion of our the thesis, we're going to cover some species delimitation of Stylaria. We want to see just how many species there are in this genus by using an integrative taxonomic approach. So we're using morphology and molecular information combined to assess the level of species diversity. And then we're going to get just a quick look at some newly created mitogenomes for 5 Naidine species with a nice mitogenomic tree, one of the largest out there currently. So with that, start with cryptic species. So what is a cryptic species? Well, a cryptic species is a species that are virtually indistinguishable morphologically from one another. So with these moths that we have over there, they're male on the left, female on the right, but going down the rows, there are 3 different species. If you look at those without really thinking about it, they all look relatively the same. I don't know if the lights need to go off to make some of the stuff easier. If not, you can leave it on. But they look relatively similar. So how do we go around kind of this problem where we have organisms that are just really similar? So we can use other evidence for speciation. So we can look at molecular divergence, see how separated they are using branches, the branch lengths. We could look at sympatry, so if these species occupy the same exact area at the same time, and we can check to see if they are reproductively isolated. And cryptic species are probably seen in just about every genus, every family, every phyla that's out there. But some of the common ones are things like Daphnia, lepidopterans, so butterflies and moths, lizards, spiders, and of course clitellates that we're going to be talking about right now. So here are our Naidids, or our Naidines, I should say, and these are our freshwater worms. So this is a pretty diverse group of freshwater annelids that consists of 2 different groups. So we have our Naidid clitellates, so those are kind of the group that we're focusing on, and then the tubophysids, which are like sludge worms, so they kind of live below the sediment. So what's cool about this group is one of the few worm species that are actually living above the sediment. When we picture of worms, we picture as organisms that are below the soil, below the sediment, but actually they are not doing that very often. They actually prefer to be above the sediment and in things like macrophytes or stones or blocks of wood. And they do have a wide range of feeding styles and adaptations to kind of help them survive in these habitats. For example, the top right photo is a Chetogastrolumni, which is an ectosymbiont of freshwater snails. So they live pretty much their entire lives in freshwater snails. And then the bottom photo is a Branchiodrillus species. Branchiodrillus are known for their really large gills that kind of help them survive and kind of clean up water a little bit. This is kind of the general Naidid body plan. This is probably the best image out there of a Naidid body plan. So the first part is your anterior end, so the head region. So we call it peristomium. Peristomium is below. A lot of Naidids do have an eye spot. There are some Naidids that don't have eye spots, but species we're looking at do have eye spots, so we'll keep that. The biggest feature of Naidids are their cheetae. So there are 5 or 6 different kinds of cheetae. Most of them have a dorsal and ventral side. The only exception is the Chetogaster, which doesn't have a dorsal. So they'll have paracheetae that we'll see a little bit later that are really kind of in Stylaria. They're very long, and they're just really pointed, sharp-looking objects with a nice serrated edge to them. And then kind of mixed in with paracheetae, we have these little small, this is kind of bumped out, little small needles that are they look about the same as the paracheetae. And then we have our ventral cheetae, and usually our ventral cheetae. And in Stylaria, these are bifid tooth structures, so they're kind of just a long point, and then at the end, they have 2 teeth, the distal tooth and the proximal tooth. And then all this kind of mess over here, those are all sexual or reproductive structures, which for Stylaria, it's really difficult to kind of see those. Man, no. Oops, wrong thing. There we go. So what group are we actually working with? We are working with Stylaria. So Stylaria is a small group, only 2 recognized species. So Stylaria lacustris, which is this individual on the right, and Stylaria fossilaris, individual on the left. What's great about this group in particular is they're one of the few Naidinids that, despite a lot of Naidinids being found above the sediment, these are one of the few that are active swimmers. So they actually will go into the open water column and swim. But they don't do that as often. They prefer to be an epiphytic species, so they attach themselves usually to plants, aquatic vegetation, or rocks, or pieces of wood. But they do have a little bit of a complex taxonomic history. The taxonomic history for these has probably been changing constantly for about 120 years. So back in the early 1900s, a guy named Michelson, he kind of looked at these, didn't really note any differences. So instead of making these two distinct species, he combined them back into one, which was just Stylaria leucostris.
Just how it is.
And it wasn't until 40 years later that we shifted them back out of that group based on the prostomium shape. Fossilaris has a big conical structure, and leucostris has that bilobate, so it's got two lobes at the anterior end. So now we're back at 2. 35 years later, we ended up going and someone ended up culturing the Stylaria leucostris, and he noticed that the bilobate feature actually changes in culture. So he noticed that it's not always bilobate, it's actually sometimes rectangular, so it'll entirely lose the bilobate structure. But we now know that that's just a cause of when they clone, they're actually just going to form that at the start, and they will eventually form their bilobate structure. And then thanks to the work of Dr. Alexandria Belli in 2005, molecular information has confirmed that there is indeed 2 species. So Stylaria leucostris, this little individual here, he's a very nice individual. He's got very nice bilobates, very nice crown, as I like to call them. They can usually get about 18 millimeters long. They're very, very small worms. Once again, he's got that nice bilobate structure. These are detritivores, so they like to eat things that are kind of dead and decaying and debris. They will eat plant material if necessary. 99% of the time, these organisms are cloning, so they are forming an exact duplicate of the parent organism by forming a little fission zone at about halfway through the body. So that fission zone, they go through this little stage where the worm kind of cuts itself in half, it forms a little proboscis coming out, and then eventually over time, it pops off and has the exact same DNA as the parent. But on the off chance that these organisms do sexually reproduce, they are usually doing it in the first 5 to 7 segments of their body where their clitellum forms. But people have kind of been able to force these organisms to change their reproductive structure. So temperature and light availability have been shown to kind of control changes in the reproductive mode, but there's some kind of combatants between the two over which is probably more right. And then these organisms are found on every single continent except Antarctica. I would be surprised if they were found in Antarctica. But most of these organisms are kind of, I'm trying to remember, Paleoarctic species. So they prefer Europe, North America, Asia, and then once you hit kind of that equator line, they're kind of like not so sure if they can survive that. But they are possibly introduced to regions like Africa, Australia, and South America. And same kind of goes for, I keep pressing the wrong thing, for Stylaria fossilaris. So Stylaria fossilaris has this nice conical prostomium that's kind of hard to see. This nice photo. They're a little bit smaller than Stylaria leucostris, so they only go about 15 millimeters. These are a pretty common prey item for insect larvae. Chironomids will eat these a lot. They go through what's called anterior regeneration, and Stylaria leucostris also possibly goes through anterior regeneration. So you cut the worm in half, it's going to regenerate at the anterior end and keep forming the head first. And then these organisms have been found, like previously, in every continent except for Antarctica. So here's a nice kind of global distribution map using GBIF data of kind of where Stylaria leucostris is, where Stylaria fossilaris is, and where both are. And as you can see, both kind of takes over pretty much a vast majority, except for Europe. Europe is kind of just dominated by S. leucostris. And there was kind of the belief that there's some boundary line, probably somewhere over here, where Stylaria fossilaris just won't go, but Stylaria leucostris will. And it's kind of the same thing for Asia. Asia originally was like all fossilaris, and and now we're starting to kind of get some Stylaria locutus in there, but there's still some debate over whether those are Stylaria fossilaris or Stylaria locutus. So now we're going to kind of go into like the big meaty portion of this thesis, which is using the integrative taxonomic approach to do species delimitation and phylogenetic analyses on Stylaria. So the main objectives we just want to ask is, how diverse really is Stylaria? Is it one species? Is it two species? Is it a million species? Well, we'll probably never know if it's a million, but it's probably not. And then we're going to kind of look at, is morphology really useful in kind of determining species in this group? So kind of our hypothesis for objective one is that originally we thought that Stylaria locutus is a cryptic species with at least two species. So we have the European sensu strictu and the North American kind of unknown. And then Stylaria fossilaris is a cryptic species comprising at least three species. So our North American being the confirmed, and then we have some from Cambodia and Japan that possibly represent two distinct species. So for our field collection and lab sorting, I would travel to field sites. I went to about 10 different states, and lo and behold, I had the worst luck ever and found Stylaria at a whopping 2 of them. So unfortunately, these worms are really small, so they're very difficult. But one of the great field sites that I have is right here on campus. They seem to dominate this little pond right on campus. So I would travel to field sites. Usually what I would do is I would collect vegetation by hand. These organisms really do like the macrophytes, especially like this Ceratophyllum season. They really like the feathery material so they can latch onto. But if no vegetation was available, I would take a dip net, sweep through for about 2 minutes, rinse the water and the net through a sieve a couple times, and finally into a jar. And then depending on how the distance between the field site to the lab, I would place the samples when I got to the lab in a white tub for ease of visibility. These organisms are kind of brownish when they go through, so they don't blend in as much in a white background. For our DNA sequencing, luckily we were able to get a ton of DNA sequences from GenBank, Dr. Christopher Sayas, Dr. Alexandra Belli, and Anatsuko Kondo. We used 2 mitochondrial and 1 nuclear loci. So we used the cytochrome oxidase subunit 1. We used the large ribosomal RNA subunit, or 16S, or the rRNL. And then we used the nuclear DNA internal transcript spacers, so ITS1, 5.8S, and ITS2. So for our alignments, I used for COX1 and ITS, I aligned them in Mesquite using MAFT with the Lindsay algorithm. For the rRNL, I used MAFT and the Kinsey alignment because the 16S rRNL has the secondary alignment structure. And then I used this great program called PhyloSuite. PhyloSuite's basically this big open source package thing that has all these things for ease of phylogenetic inference and sequence. It helps with the sequences. I'll leave it at that for now. And then within this program, we had Mr. Bayes and IQ tree that we used for phylogenetic analyses. So Mr. Bayes, we used the Bayesian inference to get the posterior probability support for our clades, and IQ tree gave us the maximum likelihood of bootstrap support. So our first little tree, for a vast majority of these, we used COX1 because COX1 has the most available sequences. So I kind of color-coded these, but I'll kind of help out since I know I didn't get time to really kind of change it to colorblind friendly. So up here, this top big block right here, these are our sensu strictu Stylaria locutus. So these are the ones from the believed type locality, or at least some of these are. The ones in this green color are from Europe, so Sweden, Italy, and Finland. The ones in blue up here are from North America, which is Canada, Washington State, and Michigan. Obviously, some of the North American ones are blending in with the European ones. These are probably introduced from Europe itself. And then down this big block right here, these are our kind of unknown S. locutus. These are the majority of the Stylaria locutus from North America. So we have Louisiana and Maryland, a whole block of Canada, and then some Kansas ones. And then towards the bottom, we have our North American Stylaria fossilaris, so our sensu strictu fossilaris. first red one over here, these are our Cambodian individuals, and then our Japan individuals. Our leucostruses were pretty well supported for each of the clades. And then when we got down to the fossilaris, the support, at least at the beginning of the clades, began to dwindle a little bit, but each little separate clade had pretty good support. This is our concatenated maximum likelihood tree with the COX-1, rRNL, and ITS data. Once again, we share the same color coding. So we have our kind of big blue block of Stylaria leucostris from North America with good support, and our big block of European individuals with good support. And then our Stylaria fossilaris, once again, not the great support, but still pretty good. And still separates into these pretty distinct clades with the Cambodian individuals kind of pairing with the Japan ones as we kind of predict, with the North American ones kind of being the weird outlier. And it should be noted that for some of these, the outgroup is Rhipistes parasita, which is the closest thing to that group, or to Stylaria. So some of the preliminary work that we did, or at least that Chris Barkow, whose work I kind of added on to, he had done some work and said that Stylaria leucostris could be 2 species. So, and this was based on phylogenetic work and very limited species delimitation techniques. And he said that Stylaria fossilaris could be 2 species based also on phylogenetic work and limited species delimitation techniques. We added the third one because we didn't have the Japan individuals for that. So what did we actually do I went backwards. So what did we do for species delimitation? So species delimitation is when we're trying to infer the boundaries between species. So we want to get those species level boundaries as opposed to the population level boundaries. So to get these boundaries, we used 5 different programs. So ABGD, the automatic barcode gap discovery. We used ASAP, assemble species by automatic partitioning, BPTP, the Bayesian Poisson tree processes, delineate, and GSDP, which is the genus species delimitation package. Before we kind of go into that, we'll just show off some of the COX-1 genetic distances between different individuals. And the overarching goal of this is that all of the average pairwise distances between the different lineages is greater than 10%, which is pretty good. But the only one that we'll kind of discover later that's kind of weird is the leucostris clade. So keep note of that. So here's ABGD. It looks like a pretty bland looking graph, but it has a good purpose. So ABGD basically uses the pairwise distance distribution to kind of see where barcode gaps are forming using sequences. And then each of these priors is kind of the barcode gap that's forming. So in the barcode gap over here is kind of like really small, like it's only like 1 species. When the barcode gap is really, really high, you wind up with like kind of this crazy amount of like 16. So with ABGD, you kind of want to find where it's kind of stable. So down over here, it's kind of stable at about 5 species. And then this kind of green boxes, and I'll start to see the green. Those are what we call the recursive. So the recursive is when maybe it's 5 species, but maybe it's 6 species, and you kind of need to test this with additional information to kind of confirm that. But with this, we're kind of staying at about 5 because 5 is kind of in our prediction radius. So for ASAP, we got 2 main things. So the only things we're really going to focus, I forgot to change this too, is the number of subsets and the ASAP score. So the number of subsets is the number of species in that partition. So there's 9 different partitions. So the ASAP score, the lower the score, the more supported that partition is. So our lowest score is only 2, and it partitions them into 4 subsets. So it partitions it in 4 species. So that's kind of matching this kind of line right here. It's really hard to see the numbers. But the first species that's giving us is this Cambodian fossilaris, only 3 individuals. The S. fossilaris is forming another group. And then every single Stylaria leucostris is 1 gigantic species. So it's kind of breaking up that idea that maybe there is a Stylaria leucostris B. And then lastly, our North American Stylaria fossilaris. Now, if we want the next highest one, we wind up at 5, and that'd be this one right here, where it splits our Stylaria leucostris in between those European and the North American individuals. So here's the BPTP analysis. So BPTP uses the branch lengths and the rates of mutation along those branch lengths to kind of propose hypothetical species. So this one, all lines in blue, everything after that is going to be what it considers to be a grouping of the same species. So for instance, the Stylaria fossilaris up here, this is our sensu strictu. The percentage is about 99%, which is really, really good. So it's pretty confident that everything after this, so this full clade, is its own distinct species. The fossilaris in Japan also has pretty high support, so it's also considering that a species. Same thing for fossilaris B. It's got a little bit lower support, so maybe it's questionable, but we'll see later. But what's kind of interesting here is our big sensu strictu S. leucostris clade. It's got the lowest score here. We know that this is Stylaria leucostris, but it does have this low score, so it's a little iffy. But what it actually does is it splits the Canada and the others into two other species. So instead of being 4 and 5, this is telling us there's 6. So it's forming this Maryland and Louisiana clade, and then all of the Canada and Kansas. So Kansas is kind of closer to Louisiana than Canada is. So I feel like maybe this could be a little inaccurate unless Kansas is just doing something a little different. So this is Delineate. Delineate is a brand new program. It took me like a year and a half to learn how to use this thing, and for a really dumb reason. But what Delineate is doing is we are telling it what species we already know, and we are asking the program to infer what we don't know. So for this one, I'm pretty confident that Stylaria leucostris Michigan and Stylaria leucostris Sweden, those are from our big sensu strictu clade. The blue represents constraints, what we know, and the yellow is the unconstrained, what we don't know, by the way. So what I did was I did those, and I'm like, well, what about the rest of these? So what it's telling me here is, yes, we know the fossilaris, and we know these two leucostrises. It is delineating these two individuals up here as two different species. So it's doing kind of as we predicted. So Stylaria fossilaris is splitting into 3 distinct species and not just a population lineages, or a population lineage, I should say. But Stylaria leucostris, on the other hand, is not delimiting into a different species. It is actually just a whole bunch of population lineages and not showing any sort of species boundary. So Stylaria leucostris for delineate is showing that maybe it's not a distinct species, but it is just a population lineage. But what happens if we change things up a little bit? So I add less populations, and as we kind of predict, things will probably change. For Stylaria fossilaris, it absolutely does not change. We still get these 2 delineated species here. But for leucostris, with less populations, now it's saying the Kansas and Canada ones are a new species, and the Louisiana and Maryland are a new species. So now it's predicting that there are 6 species here, which would fit with BPTP, but I feel a little less confident with there being less populations depicted. But it does kind of show just how hard species delimitation is and just how inaccurate it can be sometimes if you don't have enough samples to use. And then lastly, we kind of use this genius species delimitation package. We focused on 2 tests, so Rosenberg's probability of AB and Rodrigo's probability of random distinction. And so what we do is you give it a tree, a gene tree or anything, and you mark each of the clades that say automatic barcode gap discovery gave you, and you will label that and be like Stylaria fossilaris A, Stylaria fossilaris B. And what it does is for each of those clades, it shows you the species and the closest species, the probability of it being randomly distinct based on random coalescence, and the ability of reciprocal monophily based on random branching events. And what you want is you want the p-randomly distinct to be 0.05 or lower. So you don't want it to be higher. If it's 0.05, it's more likely to kind of go against the null hypothesis that they are distinct based on random coalescence, and they are more likely to be a distinct species. And if they are higher on that, then it's probably just a random coalescent event. So maybe they're not a new species. So for Fossolaris, they kind of have a unanimous decision that they're kind of pretty good, randomly distinct. But then Leucostris is kind of like, no, it's showing a little bit higher. I mean, Leucostris A to Leucostris B is practically 1, which is really, really bad. So based on P, if I use only P randomly distinct, I would feel confident, at least somewhat confident, that the Fossolaris are indeed 3 species, but Leucostris it's probably just 1 species. Now, if I only did P Rosenberg's AB, you want this value to be as low as possible. So 0.02 and lower, and all of them kind of fit that lower, especially these two, like our Leucostrises are super low. So if I only use that, I might be more inclined to be like, these are all 5 distinct species. So this is why you try and get multiple statistics to kind of verify things so you don't miss that potential error. Oops. So this is just kind of like a nice chart to kind of convey all the species limitations that we did in the changes between how many Fossolaris. They're all, for Stylaria Leucostris, however, 4 species or 2, 3, and 1 species. But with the vast majority being 1, that's kind of what we think is probably occurring, but the data is just too ambiguous to kind of show that these are actually distinct species. But for Fossolaris, we are pretty confident. So now the next question is morphology really useful in this group of Stylaria? And probably the hypothesis is there's just not enough defining characters between the European and the North American S. Leucostris to really make a really strong argument that there's a morphological distinction. So here's just a little bit basic morphology. So this kind of image in the middle, these are our Stylaria Leucostris. They're showing them the different types of prostomiums that they have. So L and M are kind of how we see Stylaria, so it's got this bilobate structure. And N and O are showing that kind of a rectangular kind of end. So recently cloned individuals. The image on the right, here's our Stylaria fossolaris. It's got the nice prostomium, the conical prostomium, I should say, and then it's Leucostris with the bilobate. You can kind of see that the cheetae, at least depicted in the image, are much smaller in Stylaria fossolaris than Leucostris. That's kind of how it goes, and if we look at the individuals, which we'll see later, between Stylaria and Leucostris, they'll look pretty much relatively the same. But what we kind of tried to see is, do these characters actually make a difference? And I don't talk too much about the proboscis, but proboscis, it's just constantly changing based on age and when they recently cloned. The types of cheetae, so we looked at the ventral and dorsal cheetae. So hairs, needles, the ventral cheetae. We looked at the genital cheetae if we could. It's very difficult to get these organisms to reproduce. So we didn't always get that, but we do have some. The hair cheetae and the needles. And then if possible, we tried to get reproductive characters. We'll talk about how difficult that was later. So for the microscopy work, what I did was I collected worms from the field or obtained slides from other institutions, and with the slides, they weren't always the best kept slides, which made morphological distinction really difficult because you would have pieces that would be breaking off, and then you were never sure if you were accurate or not with looking at those, so live individuals were always nice. I would measure the lengths of the body and the cheetae, and if possible, sexual characters at 10 to 40x. When I measured the cheetae, for example, this is not like a Leucostris cheetae, I would measure the length of the seal all the way up to the upper tooth. I would find the widest point and do the width of that cheetae. And then for the teeth, I would only do the lengths of the distal and the proximal tooth because the width is kind of negligible for at least the lower tooth. I would record kind of the minimum and the maximum length of cheetae on the certain segments. Once again, they all kind of vary, and you'll kind of see that. And then if possible, when I would get when I would get sexually reproducing individuals, I would try and stain those individuals so that we could actually get a nice detailed look at the sexual characters of these worms. which was not always easy. So this is kind of my assessment of the characters. So it's really just the lengths and the widths of the different Chidae, with the only exception being the presence or absence of genital Chidae. Some naided worms don't have genital Chidae, so it would have been nice to see if maybe that would have been a huge difference. And this just kind of shows these are more Stylaria lacustris accurate. So the hair Chidae just super long, serrated at the distal end, needles super sharp. And then the main characteristic of the ventral Chidae in Stylaria is this double-bended angular structure. So Stylaria fossilaris is kind of similar, but it only has one bend. So just to give you guys some a look at some of the at some of those. So I'll kind of describe them. So these are just some stained individuals from the North Carolina Museum of Natural Sciences. Showing some of the S. lacustris and kind of showing just how difficult it can be. So here is our anterior portion of the worm that's kind of shrunken up. And there could be Chidae up here, but there is a lot of Chidae missing from there. And this is what we're looking for. So this is our ventral Chidae. You look kind of hard enough, you can see the proximal tooth really, really small. And then the very nice long distal tooth, which is about, in Stylaria lacustris, about 2 to 3 times larger than the distal tooth. And then over here are our hair Chidae. So our hair Chidae are just kind of long. Those are usually the first things that kind of break off. They're very delicate structures. So that's where live individuals kind of come in. So here's our live individuals. Very nice and organized, with each segment having its own 2 bundles of Chidae. For Stylaria lacustris, they usually have between 1 to 3 hair Chidae and 1 to 3 needles. For Stylaria fossilaris, it's kind of the same, except they have usually 1 to 2 needles. Now, this is just showing the digestive tract of the organism. Picture B, this is once again this anterior portion of the worm, showing that it still has this kind of bilobate structure, maybe not as pronounced as the others. But one of the things that's not well documented with this species that I was able to see are these really small hairs that are up on the anterior part of the worm and up along the proboscis, at least the first half of the proboscis. There's no really a name for these kind of Chidae. We assume they're probably like a sensory organ that they use to kind of sense their environment, but there's nothing really well known about it. And then this is just a ventral Chidae that, of course, broke off during sampling. So it's got this nice double bend structure, and then leading up to the teeth. So, is it really helpful? The answer in the nice obvious term is no. There's just so much variation in ketal structures that it makes them kind of impossible to use just based on like the age of the organism, how long the organism is. They're just too diverse. But in some cases, the European worms can have larger Chidae. So the European Chidae go up to about 900 micrometers on an immature worm. On a mature worm, they can get up to about 1,000. And things overlap quite a bit. And it is possible that sexually mature individuals might have longer Chidae, but we didn't get enough individuals that are sexually mature to really look at that. So just based on like what I was able to find, there's just no obvious consistent morphological differences between these groups. There are other naided worms that are a bit more suited for morphological distinction. Things like that have gills are more distinct. They have a little bit more characters. The Stylarians, however, are pretty much confined to the proboscis, the prostomium, and the Chidae, and maybe sexual characters, if you can ever get those. So, kind of the overarching thing of this is that preliminary evidence did suggest greater speciesal diversity in Stylaria, but how much diversity did we get? The first thing that we did was Stylaria lacustris is a cryptic species comprising at least 2 species. That was our first hypothesis. But because the data is too ambiguous, I'm more inclined to think that for now, maybe it's just all one big population, different population lineages and not species lineages. So false question mark. More sequencing and sampling would help with that. For Stylaria fossilaris, we thought it was a cryptic species comprising at least 3 species. And we were pretty sure that it was these clades from North America, Cambodia, and Japan. And almost pretty much all of the species delimitation techniques said agreed with the idea that there's at least 3, so probably. And then hypothesis 3 is there's no morphological distinction between European and North American Leucoceras. And just due to intrapopulation variation and lack of characters, there's just no morphological distinction between the North American and the European S. leucoceras, unfortunately. So probably, until someone finds something, a new key data that I haven't seen yet. And this project, as Andy had said at the very beginning, was not without its challenges. So, for example, of course, the lack of morphological features, but also the staining and mounting process is a destruction for these worms. These are soft-biting organisms. So this is one that came from the North Carolina Museum of Natural History. There's like no Chidae on this one, and it's ripped in half at the anterior end, so it's removing a lot of important features. For Stylaria, all of the characters that are important are at the anterior end, and once you hit about segment 12-ish, everything really kind of repeats itself, and that's kind of where you use for genetic work. Stylaria are small and difficult to collect. Every time I go out and sample, there is always the high chance that things just kind of get out of the net or get out of this sample in some way, shape, or form. So I just never got very lucky. The only 2 places that I got it was Illinois and Kansas. So I'm going to count that as kind of at least a little lucky. And a lot of places I went to are places that they've been found in the past. So unfortunately, it just didn't work out. And then sexual reproduction was difficult to force in Stylaria. I went based off a paper that said 8 hours of daytime and 16 hours of nighttime, and they should reproduce in about 4 to 5 weeks. Yeah, I had them on that same thing, and I didn't get my first sexual reproductive organism until about 10 weeks. So maybe that has an influence of the North American individuals, maybe not. But what we really need is future directions, because this group is so understudied. There's basically nothing out there. And just to give you guys an idea, there are 195 countries around this world, including islands, and there's only 6 of them that have available sequences. So we have North America, USA and Canada, Sweden, Germany, Japan, and Cambodia. That's it. So South America, Australia, vast majority of Asia, and Africa is still left unsequenced. So we definitely need a couple more of those to kind of help global revise this group, because there's definitely a lot of stuff out there that is just kind of waiting to be discovered. And of course, these 2 putative Southeast Asian Stylaria fossillaris species, they do require a formal description so that we can get them added to the Naidid roster. So the next little part is our newly created parcel mitogenomes for 5 Naidine species that Dr. Newbig was very kind enough to help me with. I'm sure I'm missing some stuff, but hopefully I can do him proud. So for the methodology, we did DNA processing. So we wanted to amplify these 13 PCGs, so the protein coding genes, using high throughput sequencing and Illumina shotgun sequencing at the Roy J. Carver Biotechnology Center at the University of Illinois at Urbana-Champaign. So we send all our sequences out, and they would bring them back to us, and then we would fully assemble them. So we would import those sequences from Illumina with the sequence reads into Genius Prime. We wanted to map them to a reference mitogenome, particularly from Gnaeus communis, because it was the only Naidine worm available. Just to give you an idea of how little these worms are understudied. We did the de novo assembly done with medium to low sensitivity, about 25x, to make sure that all of the sequences ran pretty good. And then just to kind of confirm everything, BLAST was used to check the accuracy of the assembled genome. And then the last little bit of the work that still has to be done is I need to finish annotating some of these things, clean up some of the annotations. I haven't gotten to do that since I was working on this project, or this presentation, so I look forward to kind of trying to get that done. So it'll look nice and pretty and ready for GenBank to have them, so we can add some more Naidines. We'll pretty much double the amount of Naidines on there. So here is our mitogenomic tree that I did on the right, and a phylogenomic tree that Dr. Christopher Sayas and co did on the left. And so we try to compare the 2 trees, and the one big difference we had was this Naidid group. So our group that we're working on seems to be more closer to the polychaetes, more kind of up the tree, down the tree, I should say. While our tree was getting this kind of Chytriad group closer to the polychaetes. So that kind of changed a little bit. We also had this kind of long branch attraction in these leeches. These are our leeches. And they did have some long branch attraction, possibly due to them being more parasitic. It's like a lot. I'm trying to remember the word for that. It'll come back to me eventually. But outside of the Amphitriads, I should say, and then the Naidids, everything else kind of flows with this group. So we have our Iridinia, or Iridinia, and we have our Manila gastrids, right down here, and then our Cryptic. So we were getting a little bit different things, but it's possibly due to that long branch attraction, but it's also possibly due to just, for mitogenomics, there's just not a lot of diversity out there for worms. Our clitellates, 99% of these come from 2 different genera, so Metaphyre and Amianthus. We are missing a ton of families and a ton of genera to be represented on this tree, and if we had a little bit more sampling, we might get something that looks a little bit closer to this, as we kind of hoped. So with our mitogenome tree, it does show different relationships than a recent phylogenetic study. Possibly a difference in sampling, just not enough individuals from different families, but it also could be that long base attraction in leeches. We couldn't get the genomes to be circularized, so this AT-rich region was difficult to kind of assemble into a circular genome. And then for some reason, I did find out that some individuals are lacking a protein coding gene. Some of them are missing this ND6, which is usually right at the front. So not really sure where some of that's going, but some individuals have it and some do not. This is a one for Stylaria fossilaris from North America. So most of them know we get all these genes. We get our 13 protein protein and we get the 12S RNA, 16S RNA. So they do look very nice.
Oh my God.
And just want to give some acknowledgments out there. So of course, Dr. Anderson, who kind of watched me struggle with this project from day 1. But it was a great honor to kind of get to work on a group that you don't think you'd ever get to work with in your life, because you kind of want to focus on the big things, but the little things need some love and attention too. Dr. Charlotte Nahr and Dr. Ed Heist for willing to be on my committee, and especially Dr. Heist for willing to select it up and do some worms. Dr. Alexandria Belli and Joseph Mack at the University of Maryland for giving me a sequence for Stylaria fossilaris and Leucustris from Maryland, as well as that very nice Stylaria fossilaris photo. At the start, Dr. Christopher Arceus, who is like the leading worm guy, who I've been in constant contact with him over collection methods and mounting procedures and all this stuff, and he's been an enormous amount of help. Dr. Bronwyn Williams at the North Carolina Museum of Natural Sciences, Andy's postdoc, who, despite getting things a little late, she was amazingly helpful at providing us with some slides of Stylaria. Dr. Chris Barkow now, for kind of starting the preliminary work for this project, so I can actually finish it and make this look good. Dr. Natsuko Kondo and Dr. Akafumi Otaka at the Hirosaka University and the National Institute for Environmental Sciences in Japan. And the superb scholarship, of course, for giving money for research and for tuition stuff. And of course, the lab mates, like past and present, that kind of stuck with me through this project, watching me struggle, especially looking to a microscope for hours and hours and hours every single day. And of course, friends and family for willing to listen to worms forever and ever.